So hi guys, today I'm going to discuss about a physiology topic. It's a first year basic topic. So it's very important for you to understand this topic because you may be hearing about the isotonic saline and hypertonic saline which are used in hospitals like వెళ్ళిన saline ఇచ్చారయ్యా అంటారు. So ఆ saline ఏంటి and we need to know about the very much important topic, that is body fluid volumes and also its measurements and disturbances. So let's start with the first, body fluid volume. So first we need to know about the total body water. So what is the total body water that we have is, it is 60% of the body weight. So for all the calculations from now, we will considered 70 kg young adult or 70 kg adult man, you can say 70 kg adult man is considered as a reference with respect to either physiology or medicine. So we need to know about this, and 60% of adult men, it's nothing but total body water is 42 liters that we have. Now this 42 liters is being divided into two different groups. What is that? Two-thirds of it is in intracellular fluid volume, that is inside the cell. So if you take this as a cell, so inside the cell, this is the nucleus, so this is the water content inside the cell, that is intracellular fluid volume. So can you guess what is the next one-third that we have? Yes, the next one-third that we have is extracellular fluid volume. So this extracellular fluid volume is one-third. So you can divide it like 42, two-thirds of 42 is 28 liters and it is 14 liters. Now let us discuss about this extracellular fluid volume. This extracellular fluid volume can be again divided into three different types. I mean, it is classified or it is having these components. Number one you have is transcellular. So what is this transcellular is, we have some of the fluids like aqueous humor in the eye and synovial fluid in the joints, and also we have like pleural fluid and peritoneal fluid in the cavities, right? These are the ultrafiltrates or filtrates of plasma itself. So these are also coming under extracellular fluid volume only. So what are the transcellular fluids? Like synovial fluid, as in the joint space that we have, and also we have aqueous humor that we have, then we have pleural fluid or pericardial fluid that we have. So these all contributes to approximately 1 liter. Then we have 3/4 of this extracellular fluid volume comprises of interstitial fluid. What it comprises of? It comprises of interstitial fluid, right? 3/4 of it comprises of interstitial fluid, whereas 1/4 of it comprises the plasma. Now you can divide this 14 liters into 1 liter here, and it is 10 liters approximately, and this is 3 liters. Now let us discuss about some of the other important component of our body fluid, that is blood. So blood also comes under this body fluid component only, and it accounts about 8% of the body weight. So what is the 8% of 70 kgs is nothing but the 5 liters that we have. Out of this 5 liters, 3 liters comprises of plasma, whereas 2 liters we have formed elements or cellular components or formed elements with respect to the blood. So with respect to the blood, we have formed elements which are accounts to 2 liters. So again this plasma we have, it is divided into two different, it comprises of two different things. That is 91% of it comprises of water, whereas 9% of it comprises of solids. So these solids are also divided into, like we have 1% of it is inorganic solutes and 8% of them are organic solutes, right? So this 8% organic solutes that we have are again divided into we have 7% of them are plasma proteins. So we all know the plasma proteins, which are very much important, and they are having different roles, and the functions of plasma proteins are vast. We can discuss them in the next coming lectures. If you want, then comment down, right? So this plasma proteins that we have albumin, we have globulin, and we have albumin, globulin, and other plasma proteins also. So albumin comprises of 55%, globulin comprises of 45%. AG ratio. So when you go to the laboratory tests, ఏదైనా జ్వరం వచ్చినా లేదా any diseases like jaundice which are suspected, or liver failure, especially the liver function test, in that this AG ratio is must. So it should be 1.7:1. So any reversal of these AC ratios are also possible in different disease conditions. We'll learn about it in the further in the systemic physiology, or maybe learning it in the pathology also, right? So this is 7% of it, and 1% of it we have. This 1% is non-proteinaceous, this NPN substances, non-proteinaceous nitrogen compounds that we have. Like we have urea, uric acid. You all learned about it in biochemistry, right? You may be learning about them. So, and also creatinine. So these are the various 1% group that we have. So this is the broadest classification of body fluids in our body, right? We have first total body of 60% of the body weight is body water, 42 liters, out of which 2/3 that is intracellular fluid volume, that is 28 liters, whereas 1/3 we have 14 liters of extracellular fluid volume. Out of this extracellular fluid volume, 14 liters, 1 liter comprises of transcellular fluid, which comprises of synovial fluid, aqueous humor, as well as pleural, pericardial, all the fluids. Whereas 10 liters, the major part, 3/4 of it, comprises of interstitial fluid, which are present in the interstitium, that is surrounding the cells. Then we have a plasma, which we have, that is comprising of 1/4 of the extracellular fluid. Now we have blood, which is a other type of body fluid that we have, and it's a specialized connective tissue per se. So it is 8% of body weight. We have 5 liters approximately, out of which the 3 liters comprising of plasma, whereas 2 liters comprises of formed elements like RBC, WBC, and platelets. So this comprises of everything about the total body fluids. Now let us discuss about the measurement of this body fluids. So how to measure this body fluid? How to measurement, that is measurement of body fluids that we have. So what is the method that is used here? So the method that is used here is, it is called as dilution method, or it is also called as indicator dilution method. So what happens in this? Let us discuss in detail with respect to it. So first I'll explain you, and then I'll write here. So first thing what happens here is, you have the indicator that is used. So we should take this indicator and we have to inject it into the body fluid that we need to measure. Now the ideal characteristics of this indicator are so many points are there. I'll explain it there. So it should satisfy this ideal indicator characteristics, right? And then after it uniformly divides in this compartment that we need to measure, we need to again collect the sample from that compartment, and then we need to measure the concentration of the indicator that we have. So this is what we have in this indicator dilution method. So first I'll explain you in a simpler manner, then you'll understand it. So if you take this one as the indicator that we have, so then you take see, this is the one of the compartment that we need to measure. So this is the one of the compartment that we have, we need to measure. Now what happens, this indicator, and then this is the other compartment that we have, we need to measure. One second, I'll make it clean and green so that you can understand it in a better manner, and also you can draw in your exams also. So what happens here is, this is the one of the compartment that we have, and this is the other compartment that we have, right? So this indicator is used for both these compartments, right? So now what happens, just think logically. So this is the compartment number 1 and this is the compartment number 2. In which compartment does the concentration of the indicator will be low? I'm telling low. See, this in this first compartment it will be distributed uniformly like this, whereas in second compartment it distributes like this. It's also uniform distribution, same, but you can see the concentration here is less, whereas the concentration here we have is very much more, right? So what is the first, we'll discuss about the characteristics of the indicator. So what are the characteristics of the indicator that we have? So the first characteristic we have is, it should be easy to measure, right? We are giving this indicator to make our work easy. Again, to measure this indicator, if you are taking a hectic work, then it's no use of it, right? So we need to, it should be easy to measure. And the second point we have is, should not leave the compartment. Should not leave the compartment. So we are giving this indicator to measure the compartment and it's not staying in this compartment. So what's the use of it? So first primary is, it should be easy to measure, it should not leave the compartment, and it should not alter the water distribution in the compartment. It should not alter the water distribution in the compartment, right? And the fourth point that we have is, it should be non-toxic. Since we are measuring this fluid volumes in our body, in human bodies, so if it is toxic to humans, definitely it will be very much dangerous and we cannot follow this principle also. So it's one of the characteristics of it. And fifth one is, it should uniformly distribute. Very important, it should be uniformly distribute, right? In order to get a good results, in order to get an appropriate and good results, we need this characteristic as an important factor. Because if it is not uniformly distributed, then it will not measure the exact or apparent apparent quantity of the, or apparent volume of this compartment that we are measuring, right? So this is the important thing, and uniformly distributed. And sixth point that we have is, it should be unchanged. So while we are measuring, it should not change, right? If it is reducing or oxidizing or changing from one form to other, it is very difficult to measure. Or there is another possibility that it may be changed, but the amount that changed should be known. Amount changes must be known. So these are the characteristics of the indicator. Let's quickly revise the characteristics of the indicator that we have. First, it should be easy to measure. First point, it should not leave the container or the compartment that we have to measure. Third one, it should not alter the water distribution of the compartment. It should be non-toxic, and it should be uniformly distributed along the compartment that we have to measure. And sixth point that we have is, it should be unchanged, or if changed, the change should be known, well known, right? So then we have, so what is the formula to measure the volume? So volume is equals to I by C. So this is the formula that we have. So what is this I? So we should know the initial amount of indicator that we are injecting. So I is initial amount of indicator that we have introduced, whereas C is what we will measure. I said that we have to inject the indicator, known amount of indicator, into the compartment, and after it is uniformly distributed, we have to collect the sample, and again we have to check for the concentration of the indicator, so that the dilution, how much it happened, we are very much easily capable of understanding. So here it is the concentration of the indicator after sample collection. sorry, sample collection, yeah, something. So this is what we have another modification of this, right? If the amount, some of the amount is excreted or it is changed, so we have this I minus A by C. So what is this A here? So the I and C and V are same. What is V here? So the V is nothing but the volume of the compartment that we are measuring, right? And here we have A is nothing but amount excreted, right? So this is what we have with respect to the indicator dilution method. So you can see why it is called as dilution with this diagram easily. So here it is so much concentrated, that is the concentration is very much low. Oh, sorry, concentration is high here. Sorry, I am really sorry. Concentration is high. Here the concentration is low, right? I'm really sorry about it. So your concentration is high, whereas the volume of the compartment is low. Here the concentration is low, the volume of the compartment is high. I'm really sorry for this. It's just a matter of confusion. So for that reason only I'm doing this video, in order to avoid those confusion. So the concentration is nothing but we have the solute thing by the volume. So this is the one of the formula that we learn in your inter or also in your schooling also. So this concentration is equal to solute by volume. Here the volume, you can see it's very much high. So definitely the concentration should be low. That's what I mean to tell you. From this formula also you can see, if the concentration that we have, if it is low, then the volume of the compartment will be very much high. Likewise, we will identify or measure the volume of the compartment. Now we will see different indicators for which we use, we see different compartments, how we will measure and what are the indicators used for them, right? So we have, make it as a table here once for all. It will be very much easy for you to understand. So I can draw a table like this. 1 second. Okay. So first I'll make you clear about this. Sorry. Yeah, so I first write here the body fluid compartment that we are measuring, and next we have is the marker substances. So first we'll go with the total body water that we have. So how we measure the total body water is, we have marker substances like D2O, deuterium oxide, that is heavy water. It's very important. It's asked so many times in the MCQs and it may be coming in your university exams too or in viva. Then we have tritium oxide and also aminopyrene. So these are the three marker substances used for total body water. Now let's discuss about the extracellular fluid volume. So this extracellular fluid volume can be measured by non metabolized polysaccharides, non metabolized saccharides. So you all learn in your biochemistry about that all the polysaccharides, monosaccharides, disaccharides, sucrose, glucose, and so many things, right? In biochemistry, it's very much detailedly discussed, and this non-metabolized saccharides are first and very important thing, and it will be asked so many times. It's the best method to calculate the volume of extracellular fluid is inulin. So inulin is the best method to calculate the extracellular fluid volume, right? And also we have the chlorine, bromine, sulfate, and also we have thiocyanate. All those things which are there, these all are purely or majority they are being extracellular in distribution. That's why we'll use them. Okay. So then we have the non metabolized saccharides like mannitol and sucrose are also used for calculating the extracellular fluid volume. Right now we have one more diagram to be drawn. Sorry. Yeah, next we have third thing that we need to measure is plasma. So plasma can be measured actually. So actually I'll mention it as blood. So how do we measure blood, right? So normally we have like, generally if you go to the complete blood picture analysis, you will see the individual RBC, WBC, platelets, as well as all those parameters that we will see, like red cell distribution rate. If you have seen the CVP report, then you can understand this, right? So normally we will do all those things, and we generally doesn't employ like this, like indicator dilution method, to calculate the blood volume, right? It's and it's not necessary most of the times, right? So but we have the conventional methods that we have. So first, with respect to blood means, we have the plasma as well as red blood cells, majorly red blood cells. Actually it's nothing but formed elements like RBC, WBC, platelets, but the RBC constitutes of majority of them. That's why I have written RBC here, right? So first we'll discuss about plasma. So I have discussed about the plasma here also. So what is the most important things with respect to plasma? It is none other than the plasma proteins that we have. So we have this plasma proteins to calculate the plasma also. So let us see how. So we have radioactive iodine. So we have radioactive iodine. What is this radioactive iodine? Is nothing but is iodine 131 isotope. In pharmacology, you will learn about it in detail, that we have this radioactive iodine diagnostic uses as well as the treatment uses in the thyroid. That we'll learn in the pharmacology. So this radioactive iodine coated serum albumin as well as gamma globulin. So these are the two things that we have with respect to the plasma, how we measure plasma. And also we have Evans blue dye, which is used to measure the plasma. As well as we have this, one second, I need to make it clear. Yeah. So this RBC that we have certain substances which are penetrating the RBC membrane and tagging the RBC, right? So to find out the RBC in the blood, we need this tagging, right? So what are the substances used are, we have chromium 51. It's a very important MCQ, very important MCQ. So chromium 51 is used in tagging of RBC. Along with chromium 51, we have iron 55, 59 as well as P32. So these are responsible for tagging of RBC, right? These are responsible for tagging of RBC, right? So we can, from this we can calculate the packed cell volume or red blood cell volume, which is very easy, right? We can easily calculate the red blood cell volume, but we can calculate the plasma volume also. How is this possible? Yes, this is the another method or indirect method of plasma volume, what to say, calculation. How do we calculate this is, we have a formula that blood volume into 100 minus PCV by 100. So this is what makes the plasma volume very much clear, right? So we can calculate the plasma volume either by this radioactive coded serum albumin or gamma globulin or Evans dye, or indirectly by calculating the PCV by Winthrop's tube or Westerngen's method, that we have all those things. And then, or we have this chromium 51 which is used for tagging of RBC. So from all this we'll get the PCV, that is the packed cell volume. So from this packed cell volume also we can calculate the plasma volume. Right now, next we have is yes. So here I have somewhat cross line. Just excuse me for that. Yeah. So next volume that we are going to discuss about is, we have interstitial volume. So how do we calculate the interstitial volume that we have? It's a very much important question to be discussed. So how do we calculate? It's simple, and very simply we can calculate this with the help of a formula that we have, that is interstitial fluid volume is equals to extracellular fluid volume minus plasma volume. So just now we have known how to calculate the plasma volume. So this is the formula for interstitial fluid volume. So I have discussed, while discussing the extracellular fluid volume, I told that the extracellular fluid volume comprises of the transcellular fluid, interstitial fluid, as well as the plasma fluid, right? So this majority of them are interstitial fluid and plasma fluid. We'll just neglect this transcellular fluid for some time, right? Because it's not that much important, or just a minority purpose. It's important, but it's minority, like 1 liter only. So we just ignore it. So approximately the interstitial volume can be calculated by the extracellular fluid volume minus the plasma volume. Next we have last volume to be calculated is intracellular fluid volume. So from this detailed explanation, you can easily calculate the intracellular fluid volume. How? It's very simple. We have the total body volume, total body water that we have. We can calculate it by D2O or titanium oxide, or we have aminopyrine. We have discussed that, and we have to subtract it with the extracellular fluid volume that we have calculated using the inulin or non-metabolized saccharides like mannitol and sucrose. So with this we can calculate the intracellular fluid volume. So this completes the measurement as well as the body fluid volume as well as the measurements. We will discuss the disturbances in the next class, right? It's a very important topic to be discussed. So this is what you will understand from this, right? That's all. Thank you.